Good afternoon to everybody. It's very nice to be here with you for the first announcement of the fourth edition of the Workshop Purification Therapies that is scheduled for the end of September of the New Year's in Milano. What we would like to share with you today is the real spirit of the fourth edition of the Workshop Purification Therapy. That is the same spirit that we built in a long journey started 7 years ago. The aim from the beginning was give space for open discussion between industry, clinician, academic, in the spirit of collaborative research. That is a praxis that is like a religion for the biomedical Mirandola district. That is the main district in biomedicine in Europe. And it is at the origin of the greatest medical breakthrough. 7 years ago, we discussed how to transform idea and concrete therapeutic solution, challenging field as organ transplantation and overwhelming inflammatory disorder. This history continues 2 years later, where some of this idea became reality and several studies were launched on this basis. With the third edition, the aim was to point out the results obtaining, transforming ideas in concrete therapies, and facing a new challenge, the challenge of the evidence-based medicine. These 3 editions have been the framework to build the 2025 event. In all these years, the debates, the quality of the debates contributed to the success of the workshop, paving the way for a growing of research and for concrete application in transplantation, particularly on the ex situ organ management. I would like to underline, as 3 years ago, with the roundtable about blood purification and sepsis that you are seeing in the slide on my shoulder, we opened a real and intense debate on how to treat overwhelming inflammatory disease and how and if it is possible reach evidence in this field. I must say that we all together we contributed to start a debate that today is still central in the world of intensive care. Now, we would like to continue the discussion, starting from the achievement taken in the past years. The main question we would like to answer will be if we are really at the end of the beginning on the research and about therapeutic solution. To understand what we have understood, what are the real steps we would like to do together. We are very proud to have an outstanding scientific committee that accepts to lead this open discussion, because will be, as usual, an open discussion where all of you will be asked to participate. Now I would like to go deeper into the main themes of the workshops with the help of the scientific committee. Professor Remuthi, who supports us with his presence all this year, is going to give us a brief introduction about the result moving from research to clinic. That is the base to understand if we can actually define where we are at this moment. Please, Professor Remuthi.
I think that I would like to introduce this important meeting from research clinic on organ transplantation, and particularly on the perfusion procedure. Rather than discussing of the advantage of the perfusion procedure in a theoretical way, I like to tell you something which in my view was very transformative in organ transplantation. I mean transformative in a positive sense. Actually, this Lancet a few weeks ago reported a successful heart transplantation after 12-hour preservation with an hypothermic and oxygenated procedure. But the importance of this particular event was that heart was transported for more than 12 hours. I think it was 12.6 hours in a commercial flight. And heart was perfectly functional after transplantation. It was transplanted in a 70-year-old man who, by the way, had some important degree of renal insufficiency. Despite that, we had a perfect biventricular function after transplantation with good ejection fraction, something which represent, in my view, as the Lancet underlying a monumental advance in organ transplantation. I think that from now on, there will be the possibility of increase the number of organ transplant thanks to the infusion procedure. It will be relatively cheap because transplanting an organ in a cabin of a commercial flight means that the procedure is much more accessible to the majority of transplant centers. And the perfusion will be an essential procedure since now on in all the system of organ transplantation, and everything else will be tested on top of the perfusion procedure, which means also a complete change in our ways of organ transplantation. Everything and every advance will be tested on top of perfusion procedure, which per se has important impact in favorable modifying the following activity of a given transplantation. So be the best way asking you to be present because it will be really a new era of transplantation.
Thank you to Professor Remuthi. Now, going inside the topic of one of the topic of the event, that is transplantation and perfusion, the first question, the first thing that we have to understand is the status of the transplant activity. I think that in Italy, no one can introduce this topic better than Dr. Feltrin. Dr. Feltrin, please.
In the next September 2025, there's a great appointment waiting for us all. We are eager to meet together to discuss the results of the diffusion and the quality of donation in all Europe. This is a unique opportunity for us all to discuss and see what we can improve the quality and the quantity of the organ donations and how new technologies can support us in this pathway to improvement.
Dr. Feltrin was introducing us the discussion that we will have about the figures, the number, the achievement that we had in figures of transplantation and in clinical transplantation. Now we going into the concrete. We would like to discuss with you during the workshop where we are by clinical and technological point of view. And also if we can start to talk about regeneration and not only about conservation. Professor Gabriele Oniscu, President of the European Transplant Society and active at the forefront of this field, will introduce us on the clinical state of art. Please.
Hello, my name is Gabriel Oniscu and I'm a transplant surgeon in Karolinska Institute in Stockholm. It is a real pleasure to introduce the Aferetica webinar. We are now talking about machine perfusion technology that has been an extraordinary development in the field and has revolutionized the way we transplant. This has been driven by a significant technological development which allows us to employ a number of strategies and approaches in clinical use. Machine perfusion technology has allowed us to expand the donor pool, utilizing organs that perhaps in the past we would have discarded. This has driven the utilization and is primarily enhanced by a better assessment of organs whilst on the pump. Now, despite all this development, we still have a lot to learn and understand, and particularly how machine perfusion technology works. What are the improvements they induce in the organ? What perfusate we should use, and how should we shift through temperature from cold to warm? Machine perfusion technology, though, brings another advantage. It creates a platform for research, a platform for allowing extended organ preservation beyond the 24-48 hours, which is accepted now clinically, perhaps in 2 weeks. And in doing so, creates and gives us the ability to test new therapies to further modulate and improve the quality of the organs.
Thanking Gabriel for his presentation, now we would like to discuss about the technological state of the art. And to introduce the discussion on this theme, we have with us one of the very real pioneers in this field, the Professor Robert Porte from Rotterdam.
Dear colleagues and friends, I'm really looking forward to meeting you all and welcoming you to Milan in 2025 for the Workshop on Purification Therapies. We are going through exciting times in the field of transplantation medicine. We see a revolution in the organ preservation methods. Many devices have been developed by several companies to improve organ preservation for both livers, kidneys, lungs, and hearts, and to establish technology to assess the function of an organ prior to transplantation. These are exciting times. We are seeing a lot of these new studies and developments that can improve patient and transplant care. However, we also know that there are limitations in the technology that we are currently using. Many machines are developed for transportation, so upfront preservation, starting in a donor hospital, bringing an organ back on a machine to the transplant center, whereas in reality, most centers now prefer to use the dynamic perfusion devices back to base after static cold storage in the transplant center instead of traveling around with those machines. And if we apply machine perfusion, for example, of a liver post-ischemic after a period of static cold storage during transportation, we should probably use different kind of machines and include reoxygenation and rewarming to avoid ischemia reperfusion injury at the start of machine perfusion, if this is, for example, at body temperature. So we need versatile devices. We need devices that can apply both cold and warm perfusion, but also link the two technology together. So have controlled oxygenated rewarming as a linking phase between cold, oxygenated, and warm normal-term machine perfusion. All these topics will be discussed at the meeting in Milan, and I'm really looking forward to discussing this and meeting you all in 2025 in Milan. Thank you.
Really, thank you, Robert. Now we will approach another issue, the possibility of organ regeneration. To introduce this theme that very important and very futurable. We have Professor Mauro Rinaldi, whose experience in the field of ex vivo lung perfusion, including purification technique during treatment. And this has led to outstanding clinical results, which led him to gain the cover of ESO's newspaper. Experience that paved the way to consider perfusion as a platform for the regeneration of organs. It's really a pleasure to introduce you, Mauro Rinaldi.
Ex vivo or in situ organ perfusion has been demonstrated a very reliable platform to preserve and to evaluate graft before the implant. However, a further step forward is needed in order to achieve the real ultimate goal, which is graft reconditioning and even a proper organ repair. To do that, we need time to apply this technology during perfusion, and we need reliable evaluation tools and therapeutic strategies. And we need also to understand the mechanism more in profoundly, the mechanism of organ dysfunction before transplantation. Several potential strategies have been already experimented, but few of them are in the clinical scenario. So I do think that we have to increase our knowledge in this basic mechanism which underlie organ dysfunction to better and more precisely address the therapeutic aims. We know that inflammation is a very important issue here, and we need to address this mechanism to limit organ reperfusion injury and primary graft dysfunction. We have studied this problem as far as we know in lung transplantation, and we have demonstrated that we can fight this inflammatory response, reducing level of cytokines, of pro-inflammatory cytokines during ex vivo lung perfusion with the blood purification. And in this way, we have been able to improve the quality of graft before the transplant, and the clinical results has been really astonishing. This is one of the first attempts in a clinical setting to push ex vivo organ perfusion towards a real active and positive reconditioning. Organ quality improvement is definitely still an unmet need, and we think in the future we have to focus more and more on this subject, especially now that we are facing more compromised and more difficult donors. I'm thinking, for instance, to DCD donor. I'm thinking especially uncontrolled DCD donors. I'm thinking about patient in ECMO, which can donate organs, or patient with for lung transplantation with suspected pneumonia or aspiration. So I think this meeting will be very important to focus on these therapeutic strategies.
Thank you, Mauro. And now we are at the end of the transplantation part. And we are introducing the second big issue that we will discuss in the event. What steps have been taken for the clinical evidence on purification therapy, on the outcome of the purification therapy in critical care? We started this discussion 3 years ago. We started this discussion, but is still at the center of the debate in intensive care. This topic will be presented by Marco Ranieri and Jean-Louis Vincent, two of the actors that lead the discussion 3 years ago, and surely some of the main experts in the field of intensive care in Europe. I am convinced, really, that also the next year, this discussion will be an amazing experience as 3 years ago.
In 2022, we met in Milan to discuss the potential use, the clinical allocation of blood purification techniques. The discussion was held by an outstanding panel of clinical scientists and clinicians. And we are going to meet in 2025. What did we learn in these 3 years, and what should be the guidelines that we should follow in order to optimize the use of this technology? There is a good part and a bad part in trying to answer this question. The bad part is that, as you know, we still are dealing with conflicting data, with contradictory meta-analysis, and we still don't have precise guidelines that tell us when, how, and to whom implement this technology. On the other hand, we have learned that all the evidence that have been generated causing this uncertainty have some substantial bias. And in these years, we became aware of the importance of the concept of individualizing therapy. And this is what we should be expecting in the next meeting that in some months we will have. Elements that have been generated by clinical research and basic research that may help clinicians to individualize the therapy. Critically ill patients are much more complex than the actual available randomized controlled trial are able to study. And the most important lesson that I learned from these 3 years, from 2022 and what I see ahead in 2025, is the different way to design clinical studies and the different way to write and read guidelines. And this is what we will be doing, and this is what we'll be focusing on in our conference in 2025. How to read guidelines, how to generate evidence, and how to individualize therapy for patients that are extremely complex, too complex for the actual standard in clinical research and development of meta-analysis. Thank you very much, and looking forward to see you in Milan in 2025.
Hello. I'm pleased to address this important question of purification techniques in critically ill patients. There is no doubt about the fact that we will make progress in that direction. We know that in sepsis in particular, there is an overwhelming release of many mediators, and trying to target a single one has not been very successful. So we need to consider a way to better control the host response to a severe infection. Now, there have been many questions, and these questions are still here. Are the right patients? Probably septic shock, but when? Is it very early? Is it early? How? Which system? How to apply it? For how long? When to change the material? There are still many questions. And also, for our clinical trials, what is the endpoint? Probably not just the mortality. Mortality is influenced by too many things, including the patient preferences at the end, Outcome is determined by age, by comorbidities, by many things. So probably the endpoint will be more organ function, how we can help the patient to go over this difficult period and hopefully recover without sequela. So this is very exciting. There is a lot of work ahead of us, but we need to continue to discuss it, and hopefully that's what we will do also at that particular meeting.
With these two outstanding contributions made by Marco and Jean-Louis, we close the presentation about the central topic of the discussion we will have in Workshop 2025. Now, Angela and Fausto will give you some important information about the event. I would like only to underline that the call for abstract is open by today. That the abstract will be issued in a supplement of Transplant International and will be indexed in PubMed. Thank you to all of you. Thank you to the guys of the scientific committee. Thank you for the contribution, and I leave the stage to Angela and Fausto.
Thank you, Mauro. Hi, everybody. I hope that you are enjoying the webinar. Now, me and Fausto are going to show you some important information about the event and about the abstracts. And we are going to start with general information about the event with Fausto.
Hi, everybody. I think it's time to give some information more to our audience about the Congress. First of all, the workshop will start on September 26 in the late morning and will end on September 27 in the early afternoon, of course, 2025. What about the Congress venue? The Congress venue will be the same as usual, as per tradition, and it will be the Centro Congressi in Milan. Anyway, in the following months, new information and some details on the scientific program will be available on the official website of the Congress, such as www.purificationtherapies.com. I think it's all from my side, Angela, and I can give you again the floor.
Thank you. Now I'm going to speak about more about abstracts. Speaking about abstracts, the first thing to say is that the call is officially open. The deadline is June 2025, and all the collected abstracts will be published in an indexed supplement of the Transplant International Journal. The abstracts must be submitted in English because the English is the official language of the event, and for the detailed rules and instructions for submission, I invite you to go to our official website of the Congress. I think that it's important to say that all the submitted abstracts are going to be presented also as presentations and posters during the 2 days event in Milan. And now, for me, that's all. I leave the stage to William for the final greetings.
Thank you, Angela, and thank you all for participating today. Before closing this session, I'd like to stress out the importance for us these kind of events. In fact, all the experience and the discussion in the past edition helping us for new research project, innovative product and application. This is why I'd like to close this webinar, inviting you all to send us the abstract about your experience. Thank you, and block your agendas. See you in Milan in September 2025.